So today I want to present you my data I gained from an imaging genetic study I conducted during my PhD. Oh, sorry. Okay. On the oxytocin receptor gene and non-social decision making. So I guess all of you are familiar with oxytocin, the social neuropeptide. Just a quick reminder, it is very important in pair bonding and in pair formation, as well as in mother-pup bonding, as it was shown in several animal studies. But also in humans, the oxytocin levels are increased after social interaction, and several administration studies with oxytocin could show that emotion recognition could be increased, trusty behavior increased, and also anxiety and stress responses reduced. But most of the research is focusing the social domain and the emotional processing. And I just asked myself, how does the oxytocin system might influence the non-social domain and non-social cognition? So in the last 7 years, several studies were published showing association between oxytocin receptor gene variants and the volume of different brain areas, such as the amygdala, for instance, or the anterior insula, also structures known to be recruited in social cognition tasks. Moreover, also functional MRI studies could show association between specific oxytocin receptor gene mutations and the activation level in several regions, including the reward system as the ventral striatum. And in 2014, for me, a very important study was published by Damiano and colleagues showing that the specific oxytocin receptor polymorphism could explain some of the variants in the activation of the ventral striatum and the dorsal striatum during a non-social decision-making task. So after a quite intensive literature search, I selected 3 specific SNPs for my study, 3 SNPs which are suggested to be functional variants. The first one, the 1042778, was explored in different studies on social cognition, but also Feldman and colleagues could show in 2012 that major allele carriers would exhibit an increase in oxytocin plasma level. So for the other 2, the 237897 and 11131149, Tansei could suggest it in 2010 in a paper that the first one is associated with the regulation of the transcription of the gene, and the last one with the distribution of oxytocin receptor throughout the brain and the amygdala in specific. So for my analysis, I chose the minor carrier model. So I combined the minor allele carriers to one group, included in a full factorial analysis, as well as the participants homozygous for the major allele. This was done through 2 reasons. First, the statistical reason, the minor allele group was very small, and second, I wanted to have my results comparable to the already published literature. So I included female and male participants in my sample, so also the factor gender was included in the analysis. Just to say in the beginning, for the third SNP, I didn't find any significant differences in behavioral results or on fMRI results, so I will focus on the first 2. So as I'm interested in reward-based decision-making, I have some regions of interest. For the bottom-up activation, I focus on the ventral striatum and the ventral tegmental area, which shows an increase in activation by the presence of reward. As well as on the anterior ventral prefrontal cortex, which is suggested to control the bottom-up related activation, via functional interactions. And therefore, I applied the desire-reason dilemma paradigm, which was developed by my former department, to specifically study these regions. And just to give you a short introduction, before the actual fMRI experiment, participants learned that different colors were associated with reward. So then, in the actual experiment, the main task was to always select the target colors introduced in the beginning of the block in all of the following trials to gain a monetary reward in the end. So they had to select the target colors to reject all other colors, except there was a B letter in the beginning of the block, like a bonus round, when here now the previous learned colors were shown that they were allowed to select the additional reward. So I focused on these two trial types, one for the bottom-up related activation, when a desire was shown and you were able to select it, and the other one when a reward was shown, but it had to be rejected and the desire had to be suppressed for the top-down control. I found one behavioral difference for the first SNP I showed you, in the manner that the group homozygous for the major allele were less accurate in rejecting the reward. That means they made more errors in trials where they had to suppress the desire. So these are several reasons. There could be several reasons. For instance, that there's a stronger bottom-up related processing in this genotype group, or a disturbed top-down control. So the fMRI results for this group are shown here. I found a main effect in the ventral striatum for this SNP, and post-hoc tests revealed that this was mainly driven by the major allele group, which showed through the task more activation than the other group. And splitting into the two contrasts, so this looks highly significant, but after the correction for multiple testing, it didn't survive, and this is a very high trend, and it shows that the major allele group were not that effective in suppressing the activation during the rejection task. For this SNP, I didn't find an effect for gender, but for the next one, here I found an interaction in the VTA between genotype and genotype. And again, when I split it into the desire trials and the reason trials, you could see in the desire trials there were no significant differences, but for the reason trials, and here females with a major allele were less effective in suppressing the bottom-up related activation than males with the genotype. So there are some more oxytocin administration studies showing effects in the reward system in the ventral striatum and the VTA, that oxytocin also increase activation in non-social reward-based decision-making tasks. I just want to point out one experiment by Stripens in 2016, which found an increase in top-down control in female participants after oxytocin administration. So in contrast to me, I found like an ineffective top-down control. Just to sum up a bit, so the endogenous oxytocin system might influence non-social reward-based processing, and we find oxytocin receptors throughout the brain in the corticoamygdala circuit and the reward system. Most of the administration studies show effects in the bottom-up related activation when they're showing social stimuli. But for my two specific SNPs, I found that indications that the top-down control might be ineffective in that way. So first, this is a non-social reward-based decision-making task. Non-social stimuli were shown, and I could show modulation by the oxytocin receptor gene variations in the performance for one SNP and for both SNPs in the fMRI data. And second, my results indicate for this specific task that mainly the top-down control was more ineffective in suppressing the bottom-up related activation than the bottom-up related activation itself. But it clearly shows that we need much more oxytocin research in the non-social domain and also explore other non-social cognition. And it would be even better if we could combine the administration studies and the imaging genetic studies to see the interactions between the endogenous and the exogenous system, because there might be some differences. So unfortunately, I had my poster presentation yesterday, but I would be happy to talk to all of you. So if you see me sneaking around the conference, please just talk to me if you have comments or questions. At least I want to thank the co-authors of this work, my former supervisor, Roberto Guia-Maldonado, who is now the head in Göttingen of his lab, then my collaborators in Heidelberg, and a former colleague now in Hamburg. And now I'm doing my first postdoctoral phase in Lisbon, Indiana Prater's lab, so I'd like to greet them as well.
If there are no questions, and in the interest of time, we'll move. Oh, one quick question,
Sorry,
yes?
just one quick question. So what was the rationale to do the correction only at the SNP level, but not at the level of the RIs or the gender effect that you showed?
So the gender was already included in that model, so I didn't do any more independent analysis. For the ROIs, I did a small volume correction and not only regions of interest analysis.
So the small volume correction was applied to your whole
yes,
circuit?
to my whole brain, yes, yes.
Thank you.
Okay.